Good afternoon, morning, or evening, wherever you are. Welcome to this Shimadzu sponsored webinar. I'm Nazim Boutagu, Life Science Product Manager at Shimadzu Scientific Instruments. I will be your moderator today, and thank you for joining us. So before we start and have Simona take over, a couple notes for everyone's view and experience. The webinar console has a variety of items to help enhance your experience and interaction with us. So if you look at the screen, you'll see the following items. So on the top left is what we call a widget for questions and answers. Please submit your questions during the presentation through this widget, and we will answer them during either the Q&A session or if we run out of time after the webinar concludes, we'll reach out to you via email. Right below are the survey questions that you may fill out anytime during or after the presentation. And right across on the top right are the speaker's bio, so you may expand these items here to learn more about Simona and myself. In the center is the area for presentation slide, and right below are the resources where we added links, application notes, videos, brochures, anything relevant to the topic we are discussing today. Finally, there is a demo request form. It's located at the bottom right, and if you're interested in seeing the instrument personally or have samples to submit for evaluation, fill it out, hit send, and we will reach out to you as soon as possible to schedule. All right, now that you are a little more familiar with the viewing platform, we're pretty much ready to start. Again, if you are joining us, I'm Nazim Boutagu, your moderator. So today's webinar focuses on MALDI-based solutions for food quality control. In this webinar, we will be discussing how MALDI-TOF instruments can be used to analyze various food matrices and help detect and hence deter adulteration. Matrices food encompasses olive oil, honey, milk, and meat. Again, we ask that you submit your questions through the widget, and we will do our best to answer them at the end of the webinar during the Q&A session. We'll be starting off with the presentation from Simona Salivo, a short bio on Simona before we let her take this presentation over. So Simona was born in Reggio Calabria in Italy. She graduated in chemistry and pharmaceutical technologies and earned her PhD degree at the Department of Pharmaceutical Science and Health Products of the University of Messina, Italy. She studied under the guidance of the renowned Professor Luigi Mondello and Professor Peter Quinto Tranchida. Man, I hope I am not butchering those names. I apologize if I just did. And focused during her graduate studies on the development of multidimensional gas chromatography techniques coupled with single, quadruple, and high-resolution mass spectrometry for the characterization of lipids in food and biological matrices. So throughout her research, she developed a passion for structural characterization of lipids, which she brought into her current role at Shimadzu. So during her PhD, she was awarded a 6-month work experience at Shimadzu Corporation in Kyoto, Japan, where she worked on MALDI-TOF MS, MALDI imaging of lipids. This started her journey into the world of MALDI mass spectrometry and the Shimadzu family, where she is now a senior MALDI application specialist at Kratos Analytical in Manchester. In her job right now, she works with her team to develop numerous MALDI-based applications to showcase the Shimadzu MALDI products, and she works closely with the R&D department on the development of the Shimadzu MALDI instruments and software. She provides technical feedback and evaluation of the product at various stages of the development and manufacturing. Okay, well, enough with the intro, and without further ado, I will hand it over to Simona. Take it from here, Simona.
Thank you, Nazim, for the introduction. Hello, everyone, and welcome to this webinar. Today, I will present some examples of MALDI-TOF MS applications for food quality control. Food is important to everyone, and knowing that what we are consuming is genuine and safe is paramount. So it's key that there are analytical techniques to help ensure this. I will start with giving you some basic knowledge of MALDI-TOF mass spectrometry. MALDI-TOF mass spectrometry is a well-established and widely used technique in various fields, thanks to its simplicity and robustness. But what does MALDI mean? MALDI stands for Matrix-Assisted Laser Desorption Ionization. The process is quite straightforward. Using a laser, our compounds and the matrix are turned into an ion cloud, where they will either have a positive or negative charge. MALDI is typically coupled with time-of-flight TOF mass analyzers. The following animation illustrates the key steps of the process. The laser fires a pulse of ionizing radiation at the sample. The matrix and the sample absorb the laser energy and become ionized. A high electrical field will extract the ions into the flight tube, where they will start their journey towards the detector. Here, the ions are separated according to their mass, with the smaller ions reaching the detector first. The so-called time of flight, i.e. the time the ions will take to arrive at the detector, is recorded before being converted into a mass-to-charge ratio. MALDI-TOF mass spectrometry has several advantages. It is a soft ionization technique, as it preserves the structural integrity of the molecules, thus allowing the detection of the intact mass. It has a wide mass range, from small molecules to large proteins. It has a high tolerance to buffers and salts. It requires small sample volumes, typically between 0.5 and 1 microliter. The spectra are easy to interpret due to singly charged species being mainly formed. It is fast and easy to use and maintain. So why is food quality control important? The US FDA defines a food fraud as an economically motivated adulteration where someone intentionally leaves out, takes out, or substitutes a valuable ingredient or part of a food. It also occurs when someone adds a substance to a food to make it appear better or of greater value. Adulteration is by far the most popular type of fraud and can occur as replacement of a nutrient, ingredient, or part of the food with others of lower value. Dilution, where high and low value ingredients are used at the same time. Unauthorized addition of unapproved ingredients to improve or disguise the quality of the product. Concealment of low quality of food ingredients. Food quality control is also important to determine authenticity, confirm the geographic origin, and to assess toxicity. So these are the applications that I'm going to discuss today. For 3 of these, applications news are available. So if you're interested in learning more, you can contact your local Shimadzu representative. 2 applications were presented at the last ASMS 2023 conference and will be soon available as application news. So starting with the first application, this is about how we can use MALDI-TOF MS combined with multivariate analysis and machine learning to tackle the issue of extra virgin olive oil adulteration. An application news is available for this. Extra virgin olive oil is the highest grade and least processed form of olive oil due to its unique production process in which olive fruits are crushed solely through mechanical pressing under cold conditions, thus preserving its full flavor profile, characteristic, and nutritional properties. EVO has also been recognized as being nutraceutic, as it provides nutritional and health benefits, thanks to its optimal balance of saturated, monounsaturated, and polyunsaturated fatty acids, and minor components such as polyphenols, tocopherols, and sterols. Some research suggests that EVO could protect against heart disease, combat cancer, and inflammation. Due to the high value of EVO, Adulteration has become an important issue in the industry, with adulteration by addition of cheaper vegetable oils being very popular. Besides the economic damage, there are also health and safety concerns due to the exposure to potentially hazardous allergens, such as those in hazelnut oil. To combat Evo frauds, rigorous and efficient quality control assays have been established to protect the identity of this unique product. Approved chromatographic analytical methods based on triacylglycerols and sterol profiles by HPLC and GC are quite popular. However, these methods often have the drawback of requiring time-consuming extractions, large solvent volumes, and long analysis time. Therefore, it is becoming increasingly important to simplify and speed up these analysis methods, while at the same time guaranteeing the quality and robustness of the data. Triacylglycerols account for more than 90% of oils. The tag composition is unique for each type of oil, as the fatty acid expression is plant-specific. In this application, we propose a simple and powerful analytical method to tackle the problem of Evo fraud, using MALDI-TOF MS combined with multivariate analysis and machine learning. So here is the analytical workflow. It consists of a minimal sample preparation, as it does not require any extraction, but only a simple dilution of the oil in solvent. The adulteration was simulated by spiking the Evo with sunflower oil in quantities of 5, 10, and 20%. The MALDI analysis can be performed and automated on a MALDI 8020 or 8030 without the use of a MALDI matrix, therefore by simply applying the diluted oil directly onto the MALDI target. Follows the multivariate PCA and machine learning analysis with the Shimadzu EMStat solution software for the classification of unknown data. So here we have, from bottom to top, an example of tag profiles of pure Evo, Evo adulterated with 10% sunflower oil and pure sunflower oil, all obtained with the matrix-free LDI analysis. Although the tag profiles of pure Evo and sunflower oils are very different, it is clear that it is not possible to easily distinguish a genuine Evo from an adulterated one, just with the naked eye. The addition of the sunflower oil, even in small amounts, can alter the tag ratios. However, these subtle differences will not be noticeable by a simple look at the tag profiles. So here is where the statistical analysis can help us. At first, multivariate analysis was carried out by principal component analysis. We were able to obtain a good separation of the three groups, as you can see in the PCA score plot on the left. The PCA loading plot suggested various potential markers that contribute to the separation, so that points which are further away from the origin greatly contribute to the separation, and also their position on the plot suggests which group they are more important. For example, the marker at M of Z 901 is located in the left-hand side of the plot, hence it's highly representative of a sunflower oil. And in fact, the ROC curve of this marker demonstrates its statistical strength, with values of sensitivity and specificity of 100% and an area under the curve of 1 with respect to Evo. The second application I will present today is on the use for the first time of MALDI-TOF MS to determine the authenticity of Manuka honey. This work was presented at the ASMS 2023 conference. Manuka honey is a unique type of honey that is produced by bees that pollinate the flowers of the Leptospermum scoparium bush, a tree native to New Zealand. Manuka is somewhat different to other honeys, as it has reported health benefits and qualities such as antimicrobial activity, antioxidant, and anti-inflammatory properties. In fact, today it is increasingly used as a natural home remedy to fight bacterial infections. Over the centuries, it has been widely used to promote wound healing. as there are strict criteria that must be met in order for a honey product to qualify as Manuka, Manuka honey attracts a price premium, making it an obvious target for adulteration and misrepresentation. Examples of Manuka honey frauds are blending with sugars and syrups, mixing with cheap honey alternatives, or mislabeling of the floral and geographical origin. The New Zealand Ministry of Primary Industry has approved a variety of genetic and chemical tests to certify the authenticity of the Manuka pollen. The chemical tests are based on chromatographic methods to detect the unique markers, among which is the leptospirin, a known marker for authenticity. Leptospirin is used to attribute the unique Manuka factor grade. In this application, we demonstrated for the first time the use of MALDI-TOF MS to quickly screen Manuka from non-Manuka honeys through the detection of the unique markers. The analytical method is simple and fast. The sample preparation consists of extracting the polyphenols from the honey with solid phase extraction with a reverse phase polymeric sorbent, in which the polar components, such as the sugars, are not retained and eliminated with the aqueous phase, whereas the hydrophobic components, such as the polyphenols, are retained through hydrophobic interactions with the sorbent and then eluted with a polar solvent, such as acidified methanol. The removal step of the sugars is very important, as these are generally the most abundant components of honey and therefore would mask the minor marker components. The polyphenolic extracts were analyzed with the MALDI 8030 in positive ion mode and THAP matrix. The identity of the polyphenols was confirmed by MSMS. Here we have the MALDI profiles of the polyphenols extracted from the honey. We can see in the Manuka honey profile how several markers are detected, including the leptospirin, while these are not detected in the non-Manuka honey. For the identification of the markers, we use MALDI MSMS and compare the fragment profiles of the commercial standard and the sample, as we can see in the example on the left of the 3-phenylactic acid marker. For the leptospirin, unfortunately, there are no commercial standards, but since its structure is known, it was still possible to identify it by interpreting the peaks of the fragments. The third application I will present today is about synthetic food colorants in confectionery products. So let's see how MALDI-TOF MS can be useful. An application news is available for this application. Synthetic colorants are a type of additive that are widely used in food, pharmaceutical, and cosmetic manufacturing. Among these, sulfonated azo dyes are popular because of their stability, solubility in water, and low cost. In food, colorants can be used to make food more attractive and appetizing, provide or enhance colors already present, or correct natural variations in color. Colorants are subject to stringent regulations by different bodies around the world over their safety and adverse effects on human health. For example, in the United States, the FDA is responsible for the approval of colorants for use in foods, drugs, and cosmetics, while in Europe they are regulated by the European Food Safety Authority. Among the various food colorants, there are some which have been flagged following scientific research over a possible link to attention deficit hyperactivity disorders in children. It's the case of Sunset Yellow SCF, Tartrazine, and Allura Red AC. In the US, the FDA has pursued no action on banning these dyes, as the evidence was deemed inconclusive. In the EU, instead, while these dyes have not been completely banned, food manufacturers are required to apply warning labels on products containing the dyes of concern. Here we show the capability of MALDI-TOF MS to detect sulfonated azo colorants in commercial sweets known to contain these colorants. The analytical method is simple and fast. The extraction of the azo dyes from the food product was performed by ion pair extraction with BTAB cation surfactant. The BTAB and dye ion pairs were then extracted in chloroform and separated from the aqueous phase. The extracted dyes were analyzed on the Shimadzu MALDI-8030 with 9AA matrix in negative ion mode. So the method was first developed and validated with the standard dyes. Here on the left is the MALDI spectrum of the sunset yellow FCF dye. You can see that the expected M of Z of the dye was detected with good mass resolution and accuracy. Then we proceeded with extracting the azo dyes from the commercial candies. Here on the right, we have examples of MALDI spectra of each colored product, where all the dyes detected are consistent with its color. For example, the green candy contains the yellow tafrazine E102 dye and the blue brilliant blue FCF E133 dye. In the fourth application of this webinar, I'm going to show you how MALDI-TOF MS combined with multivariate analysis can be used to differentiate different types of meat to pinpoint a meat adulteration. An application news is available for this application. Adulteration in raw meat can occur in the form of partial or total substitution with another cheaper type that may not be fit for human consumption. We probably remember the horse meat scandal of 2013, which was big news in Europe, where Findus products containing beef were removed from the market as tests revealed the presence of horse meat in quantities from 60 up to 100%. In 2021, the horse meat scandal resurfaced, with reports of horse meat sold as veal. This type of criminal activity highlights a complete breakdown of traceability and quality standards in our food chain. In this application, we will see how MALDI-TOF MS combined with multivariate analysis can help in the screening of raw meats to confirm the animal species of origin. The analytical method is simple and fast. The sample preparation is minimal, and it consists of a microscale extraction of the proteins by a simple homogenization of the meat in acidified water. The protein aqueous extract is then analyzed on the benchtop MALDI-8030 instrument, followed by multivariate analysis with the Shimadzu EMStat solution software. The pure meats used to obtain the differentiation were beef, lamb, chicken, and pork, while the adulterations were simulated by spiking a certain type of meat with another one in quantities from 25 to 75%. Here on the left, we can see the MALDI protein profiles of the various pure meats. It is pretty obvious that they are quite different. In fact, this has allowed a very good separation of the classes with the PCA analysis. Not only the classes of the pure meats are well separated, but also the 50% mixtures, which are positioned in between the corresponding pure meats. To give you an example, the binary chicken and lamb mixture is located between the pure chicken and lamb meats. Now on to the last application. This is about using MALDI-TOF MS to differentiate A2 cow milk from mixed A1-A2. This work was presented at the ASMS 2023 conference. So why is A2 milk important? About 10,000 years ago, all cows were producing milk with A2 beta-casein. However, a genetic mutation affected European cows, prompting them to produce milk with A1 beta-casein. The human body, which was able to easily absorb A2 milk, has not yet fully adapted to metabolize A1 milk, which is why side effects may occur. The mutation involved the gene coding for the amino acid in position 67 of the beta-casein, where the A2 variant has proline, while the A1 variant has histidine. Regular cow milk contains a mixture of A1 and A2 beta-casein, while A2 cow milk is produced by cows that naturally produce only A2 beta-casein. According to several studies, A2 milk has various health benefits, such as a better digestibility. Some states, such as Australia, New Zealand, China, and the United States, have supported these studies, and A2 milk is commercialized. The European Union is still skeptical about the reliability of these studies. However, nowadays, more European states have started to commercialize A2 milks. For all these reasons, A2 milk has a greater economic value, and therefore tests that can differentiate A1 A2 milk from A2 only are important in quality control. Currently, only genetic tests are available. In this application, we demonstrated how MALDI-TOF MS can be useful to differentiate A1 A2 milk from A2 only. The analytical method consists of performing an enzymatic digestion with trypsin of the protein component of the milk. Two trypsin digestion methods have been developed. The first is the classic in-solution method with overnight incubation at 37 degrees, and the second is performed directly on the MALDI target, and the incubation time is only 15 minutes. The protein tryptid digest was then analyzed with the MALDI 8020. We can see from the results of the in-solution digestion how in the A1 A2 milk, both peptides coming from the A1 and A2 beta-casein variants are detected, whereas in the A2 milk, as expected, only the peptide from the A2 beta-casein variant is present. And we get similar results with the on-plate digestion, and this with a digestion time of only 15 minutes. Finally, the limits of detection were evaluated for milk mixtures containing various amounts of A1 milk in A2, for example, from 1 to 50%. Two MALDI matrices were evaluated, i.e. synapinic acid on the left and synapinic acid ATHP mixture on the right. We can see how the LODs were improved with the synapinic acid ATHP mixture, as this allowed the detection of A1 milk in A2 down to 1%, as opposed to the synapinic acid alone, where the presence of A1 milk in A2 was detected only down to 10%. In conclusion, MALDI-TOF mass spectrometry is a well-established and widely used technique in various fields. Compared to other analytical techniques, it stands out for its speed of analysis, simplicity, and robustness. In this webinar, we presented various MALDI-TOF applications for food analysis, in which simple and rapid methods aiming at combating the problem of food fraud were developed. I hope you found this webinar informative. If you're interested in learning more about the applications presented, please contact your local Shimadzu representative. Let me end by thanking all of you for being here today and for your attention. Now, back on to you, Nazim.
Thank you, Simona, for such an insightful presentation. I would like to also thank our audience for attending and sending questions. I think we have time for a couple of questions, and we'll answer some of them. Simona, are you ready?
Yes, Nazim. Yep.
Great. All right. So let me look through the list here. How about, I actually noticed this one. I was like, oh, we could start with it because it's kind of a basic question on the MALDI operation. And so it says basically, how long does it take to generate a MALDI-TOF spectrum for one sample?
Well, it depends. So a MALDI spectrum is typically the result of the accumulation of a certain number of laser shots, but it also depends on the concentration of the sample, how pure is the sample, and how easily it ionizes. However, in general, it can take from a few seconds to a couple of minutes, so it is quick. Of course, this is the case for regular non-imaging samples, because with imaging, it can take from several minutes to hours, obviously, depending on the size of the imaging sample. Yeah.
Yeah, I'm glad you brought up imaging, and since we've talked about our benchtop, and it's known as an entry-level MALDI-TOF system, being like a benchtop linear system, it's Shimadzu really is the only vendor to offer an imaging option to be carried out on the system. So a very, very neat niche application. All right, so continuing with questions. Yep, and I think this one is also basic, but like can translate into the application itself if you want to go really implement those methods. And it's related to the automation, and it says, can the data acquisition be automated, e.g. for larger sample numbers? Meaning, like if you have a lot of samples, do you have to be there and manually record each spot, or can you just create a list and run it?
That's a very good question, actually. Oh, no, absolutely. We can automate large sample numbers. We can do this in the main acquisition software, or we have a dedicated automation software that just simplifies the acquisition of large sample numbers when, for example, you're using the same method. We also have a specialized automation software, but that's for targeted QC analysis. We also have some software tools that can be used to improve the quality of the acquired data. So basically, this is by setting some minimum requirements, and then those texture which do not meet these requirements will be discarded. So yeah, there are a couple of options to automate a large sample numbers. Yes.
Just to add to this answer, some of the sample prep require, for instance, some SBE cleaning, and I believe these methods also, when you have like a large sample number, can be automated using liquid handlers. So does the multi-plate spawning. All right.
Yeah,
so continuing with the. Go ahead. Sorry, Simone. I moved on.
I'm sorry, I was just saying, I suppose, yeah, there are ways also, to automate, the sample prep, side, but definitely on the software side, definitely we have automation solutions for, to acquire, a lot of, samples, yeah, definitely.
So we do provide now 2 different sizes of plates, right, with the FlexiFocus plates that were released.
Yes, correct. We have the standard 48 well format, and now we have also a higher density one, which where you can spot up to 96. You can analyze up to 96 samples. Yes, that's correct. Yeah.
Outstanding. This is going to help greatly with the throughput. Yeah, moving on into the application, we have one question regarding the milk application, and the question reads, regarding the milk application, why was it necessary to digest the sample prior to analysis? Could you not detect the difference by analyzing the intact proteins? Simona, I'll take this answer. Because actually the note was developed by my group. So the A1 and A2 beta-casein whole proteins are challenging in 2 ways when you analyze them intact. Once the first challenge we met is once we crushed the milk protein by adding acid, it was fairly difficult to resolubilize them, and the sensitivity of the entire of the protein detection was low. And the second major hurdle, to analyzing the intact proteins was at 23 kilodalton, which is the weight of the molecular weight of the beta-casein protein. It was difficult for us to resolve properly the 40 Dalton difference that will show A1, which is the molecular weight difference between the A1 and A2. So digesting was the way to go because the resolubilization was just easier. And then once it is digested, the detection at 5, in the lower mass range, and in our case it was around 5 kilodalton. We have way enough mass resolving power to separate the 40 dalton mass shift between the two peaks of interest. And the digestion also enabled us to increase the sensitivity and being able to tackle low mixtures and being able, I think we went down to less than 5% A2 into the A1 milk, if I'm correct, right? If I'm reading that, if I remember this correctly. Yeah, so the challenge also with the application, since we introduced the digest, the proteolytic digest, it was to make it amenable for high throughput, and this, thanks to the team's hard work, we were able to create like an on-plate digest that lasted only 15 minutes, very easy. And the whole sample prep actually came back into a more manageable time. They enable implementation into a higher throughput type of setting. Yep, it was a bit of a long answer. Hopefully I didn't lose our audience here, but yeah, feel free also to keep asking more questions on top. Via the chat, and also send us emails, and we would love to keep discussing those applications specifically with you guys. All right, moving on. How quick and easy it is to screen for the Manuka-Honey authenticity marker on your entry-level system?
Right. I would say the sample preparation takes the most time, although it's just a simple SP extraction and it only requires very, very small volumes of sample and solvents. But once you have the polyphenols extract, the actual MALDI analysis can take, again, a few to several seconds. So yeah, again, it depends on the sample, on the laser shots and on various things. But yeah, in general, yeah, it's still very, very, very quick. Yeah.
And continuing with the Manuka honey, we have another question, and it reads, can the Manuka honey method be used for other types of honey, like clover honey, orange blossom?
Definitely. The SP extraction is not specific to the markers of Manuka honey, but it can extract any kind of polyphenols. I mean, at the end of the day, the Manuka markers are polyphenols. So I guess if you know specific markers of a certain type of honey, then yes, this method should work. Although we only tested it for Manuka honey, but yeah, in principle it should work. Yes.
Thank you, Simona. All right, looking again. Probably it would be good to bring up the microbial ID in the discussion. So one question said, can you perform microbial identification on our MALDI 8020, 8030s?
Yeah, I mean, in principle, you can perform microbial analysis on the MALDI 8000 benchtop system, but unfortunately, we don't currently have a validated database of reference data that were acquired on the benchtop instruments, which obviously would be used for the identification. But for microbial identification for this application specifically, we offer the Axima ID Plus series of instruments, which are robust, they are well-established platforms, and they are coupled with the Saramis database. So yeah, at present, unfortunately, we cannot provide, a microbial identification application for the benchtop systems, but yeah, again, we have the Axima ID Plus instruments for that. I don't know whether you want to add something, Nazim, on this topic, but yeah.
Oh yes, definitely. And I like the question because I believe that our audience, especially if they're specialized in food testing, a lot of them have come across MALDI-TOF through the use of microbial identification, and I believe also that what we're showing here, which is another dimension to MALDI-TOF that would be very helpful to them. And I did welcome all the work that you've done, all throughout, and I was actually very excited in putting these applications out on the webinar because it will show users that of an RUO, for instance, like an RUO microbial ID platform such as the one we sell, the number of applications they can also implement on the same platform. So, just said shortly, basically, if you purchase an RUO microbial ID platform, you can of course run microbial identification, but you can also do your honey testing on it, your A1, A2 milk differentiation, your milk, your meat typing, and etc. So there's a lot of value from the MALDI they will be purchasing, and microbial ID actually has like a great return on investment because of the throughput and ease of use. But you add those, and if you add more of these tests, I mean, the ROI will get like even shorter. So a great, flexible, high-value platform that we offer. That's pretty much it, Simona, for the questions. Thank you again for making yourself available to narrate those slides and actually show us a little bit what MALDI can do for food quality control.
No worries, Nassim. My pleasure. Yeah.
Looks like we are at the end of our allocated time for the webinar. Thank you all for the interesting questions. Since we don't have time to answer all of them, we will reach out individually to each one of you whose questions we were not able to answer today. Once again, thank you all for attending. We will send you an email with a link to view a recorded version of this presentation, and you can vision it anytime. We hope to have you back in webinars we organize in the future. Have a great day.